So welcome everyone, and thank you for joining us today for our webinar titled Turn Away from Traditional Tethering and Towards a Better Method for Data Collection. This is Haley Kellerton from Inside Scientific, and I will be your host for today's event. Our session is sponsored by BASI and will feature experts discussing the use of a novel movement-responsive rodent caging system as a means to minimize animal stress and enable unique discovery in many research applications, namely neuroscience, animal behavior, drug discovery, and cardiometabolic disease. First, we will hear from Candace Rohde-Johnson, Director of In Vivo Products and Services at BASI. Candace has worked with clients worldwide to provide training, seminars, and workshops focused on improving research results without sacrificing animal welfare. And today, she will discuss how tethering systems can be used as a hands-off approach to data collection for research applications including microdialysis, open flow perfusion, telemetry, and optogenetics. Following, we will hear from Dr. John Cirrito, a leading Alzheimer's disease researcher and professor at Washington University of St. Louis. He will present an overview of the data his team has collected through an in vivo microdialysis technique that enables minute-to-minute measurement of interstitial fluid, AB, within the brains of awake and behaving mice. He will also share additional study applications made possible by using the Raturn system.
Great. Thank you for getting us started, Haley, and welcome everybody. We're going to talk today about turning away from traditional tethering and getting better data. So I'm going to give you the overview of the system so everybody has a better understanding of exactly what it is and what it does before Dr. Cirrito takes over and tells you a little bit more in depth about what it can do. Our Raturn system is a replacement for a traditional tethering system. It doesn't require a liquid swivel. It doesn't require a commutator. The caging moves in response to the animal. And because of the way that it approaches this problem, it creates an extremely flexible system for small and for some large animal models. We find that it's very difficult to explain to people exactly how the system works. And so instead, what we're going to do is show you a brief video so you can see it in action and have a better understanding of how it all comes together. Great. So as you could see there, the system for small animals works for both rats and mice. We also have had people that have used hamsters, guinea pigs, on occasion chinchillas. And by far and away, our small animal system is the most popular. We have a couple of different configurations available, but the base components of the system include a turntable and platter, an upright arm to hold the balance arm, a motor and controller so you can turn it on and off. We have a couple of different ways that you can put it into your lab depending on what you need. So there's a simple standalone unit. There's also a cube-shaped unit, which you can see in the bigger picture up top with the cute little stuffed rat in it. And that one allows you to get your instruments closer to the animal. So if you're working with anything that needs shorter lead lines, a smaller length of tubing because you are worried about sticking or something in your fluid lines, then that is an excellent way to address it. The systems can also be modified so that you can do metabolite collections. So urine and feces collection. You would need a special cage in order to make sure that that happens. And the caging component is the final part of all of this. We have a couple of options available depending on what you're trying to do and with what species. We also have a large animal system, which you can see on the right-hand side. It was developed specifically for use in swine models. And essentially just built on what we're going to talk about as we move forward today, the benefits of having a system that responds to the animal's movement instead of working with a traditional swivel or commutator. So to understand why it's better, you have to have a pretty good understanding of what the alternatives are, and we'll start with the liquid swivel. So a liquid swivel is a simple way to connect fluid lines, and it works with internal seals that maintain the connection from top to bottom. The good things about a liquid swivel are that they're relatively low cost. They're great for single fluid lines, relatively good for up to 2 fluid lines. But when you get past that, you have a couple of things going on. One is that it becomes increasingly difficult to turn the mechanism. And so when you're working with something like a mouse, it can be very hard to have a system that can accommodate all you want to do and still will respond to the animal movement. The second is that you're not able to connect it up to a commutator or an electrical line at the same time. You're usually pretty limited here. So that means that it is best for single or dual infusion or sampling. It can be used for microdialysis and OFM, open flow microperfusion, if you've got a dual channel swivel, but it cannot be used with simultaneous electrical lines or cables. The next option is a commutator. This is sometimes also called a rotary joint, a signal transducer, a patch cord or patch cable. That kind of depends on what industry you're in, but ultimately what it is a signal relay for electrical or optical signals. It works with multiple contact points that help relay the signal from one part of the cord to the next part of the cord, allowing the animal to move around freely. So the pros on this is that it's great for applications involving only wired leads. It's often provided as an option from the instrument supplier where you're getting your optogenetics or EEG recording system or whatever system it is that you're trying to work with. The downside is that you cannot combine it with fluid lines. You can typically only transfer specific types of electrical signals. So if you had a commutator for EEG, it almost certainly would not work. It wouldn't work as a patch cord for vibrophotometry, right? So you're limited. You would have to have it for each of the systems that you're using. And so what that means is that overall it is best for wired leads specific to an instrument and usually cannot be combined with fluid sampling. And then there's the Raturn. So as you've seen from the video, this is the movement responsive caging. It provides direct connection from your animal to your instrument of multiple sampling lines or cables to the awake, freely moving animal. So all 3 of these systems let your animals move around the cage in the way that they want. But the Raturn is the only one that has the direct connection. So you're not having tubing connectors, you're not having those commutators, you're not having to worry about you're connecting from animal to swivel to pump. It's just one straight connection, which minimizes both the volumes and also the potential for disconnections. The tethered animals activate a sensor that rotates the cage when it's needed, and that's what allows you to connect, to maintain rather, that direct connection. It also has the added benefit of allowing us to collect activity data. The pros on this are that it's a flexible system. You can put a lot of different types of connections into your animal. It's low stress and it allows you to combine fluid and electrical leads as well as optical cables in a single rat or mouse, as you'll see in just a moment. The downside is it's not necessary for simple infusion studies. If you were just doing a single infusion line, this maybe isn't the instrument for you. It would work just great for that. It would allow you to kind of build your study and do more, but it's not always going to be the perfect solution. But this system is the most flexible system for any type of study. So if you're trying to combine different types of sampling applications, if you're trying to start small but build from there, then this would be perfect. And because you can add in the activity monitoring, that gives you just an added benefit. So the first question that everybody has when they see the system, and you probably had it yourself when you were watching the video, is does the rat get dizzy? So I do not have data on whether the rat gets dizzy or not, but I do have a lot of data about how they do acclimate to the system and how long it takes for them to return to baseline levels. When we started with this system, it was originally created for microdialysis. And so, of course, we were interested in neurotransmitter release in the brain. So we looked at microdialysates. We have developed the system for use in automated blood sampling applications. And so we also have data on corticosterone in the bloodstream in rats. And then we also have the basic activity data looking at how long it takes them to return to baseline levels. So for all of those parameters, the kind of acclimation, return to baseline levels tend to be somewhere between 2 to 4 hours for the animal to return to where you would expect them to be. However, sometimes we're doing something more stress sensitive than that. And so this is a great example of the slide that you see in front of you. I know it has a lot of information here, but the basics are this. We had a team a client that was using our automated blood sampling system so that they could combine automated blood sampling, dosing, and telemetry to reduce the total number of animals that they were using for their safety pharmacology studies and to get better data from each animal on the study. They wanted to do this, but of course, in order to make it happen, they had to make sure that changing the system, the platform on which all of the sampling was done, wasn't going to change the results of the study. So they set forth in a wide variety of testing to make sure that everything was going to behave the same. So here you see these very stress-sensitive parameters: arterial blood pressure, systolic and diastolic, heart rate, and body temperature. These things are well known to be affected by just a human walking into the room, and you need them to be very clear for your studies when you're looking at changes to these parameters based on dosing of a drug. So what they did was compare the ABST, which in this case means automated blood sampling plus telemetry, so that is the Raturn-based system, with home cage telemetry, which means the animal was in a regular shoebox cage and was tethered with a standard stainless steel spring swivel. And what you can see from the results is that there was an acclimation period time. It's higher than that 2 to 4 hours that we see for most of the parameters, but it is identical for the home cage versus the Raturn cage. Okay, so this data indicates that there is no difference between putting an animal in the turning cage and putting them in a clean home cage for a new experiment. The other thing I like about this is that we don't always think about the impact that just putting an animal in a clean cage has. Most people wouldn't have thought about the fact that putting an animal into a clean home cage would have a 3 to 5 day acclimation period for them to get back to feeling like it was home. So interesting side note on that. All of this was published. You can see the reference there at the bottom, and you can see the other indicators that they looked at for this, but we are going to actually look at one more here, which is that beyond just the acclimation period, they wanted to see how sampling method influences stress and hormone release. So this is where you start to bring the human element into it. So in this case, we had still the home cage scenario. So same thing, an animal in a home cage with a swivel and a coiled tether. The ABST, which is the Raturn system with automated blood sampling, and a tail bleed. So that was the animal in a restrainer with a tail nick to take the samples. So obviously, the tail bleed is a much more hands-on approach. You would expect there to be higher levels of stress hormone release in those tail bleed animals, which you can see for the corticosterone, insulin, and prolactin. What was a little bit unexpected is that the ABST had lower stress hormone release for those same compounds as the home versus the home cage. And so just to clarify, the home cage is not touching the animal for that blood sample, but a human had to come into the room to take the sample from outside of the cage. So the swivel had a connection piece where they were able to connect a syringe and take the sample without touching the animal. And this shows that just the presence of a human in the room changes the stress hormone release in animals. So for a lot of our clients who are using the system, they're using it for automated blood sampling, and they say, well, I don't care about stress hormones. Of course, that depends on exactly what you're studying. But in the case of pharmacokinetics, for looking at new compounds of interest and taking it to market, that is what a lot of our customers are doing with this system when they combine it with our automated blood sampling system. And so they think they don't care about the stress hormone release, but people don't always think about what also happens when you stress an animal out. So this looked at automated dosing versus manual dosing. So this means that while they're on the system, they were either picked up to be given a gavage dose, that's the manual, or they were given a dose through a programmed pump into a stomach catheter. So in this crossover study, you could see a very distinct difference in the pharmacokinetics and the drug uptake for nicotine in this case, that was based on the method of dosing the animal. So we know that the method of our intervention with the animals, the way that we design our study, can impact results, and this is a very good example of that. So you have a basic understanding of the system and an understanding of why we think it's important to be able to get your hands off of the animal so that you can get that better data. And now we'll talk a little bit more about all of the possibilities that you can explore with the Raturn. So starting from the left bottom there, our little turquoise bubble, you can see that we have activity data. So our system is not a behavioral collection system, but because the animal is in there and is triggering a sensor to make the cage counter rotate, you're able to collect basic activity. Where this is most helpful is in looking at things like pharmacodynamic response. So you give a drug and you look at what happens in response to that administration of the drug as a result of the dosing level. Another area where it's very useful is for circadian rhythm studies. Going from there, we have an amazing system for optogenetics and vibrophotometry because you don't have to have a patch cord, you don't have to have any kind of commutator or transducer. Everything is a direct connection, and so you have less signal dropout. And of course, remember that you can combine that with the fluid sampling. We also, as I mentioned, started with this system for microdialysates, interstitial fluid sampling. So microdialysates or open flow microperfusion, which is an approach for taking large molecule sampling that's very similar to microdialysis in terms of perfusing through, but those are possible. You heard me mention automated dosing and sampling of blood. So intravenous dosing and sampling is easy to be done manually, or you can combine it with an automated system. Our Kulex system is what you saw referenced in the slides for stress here. And finally, you can also do things like physiological monitoring, so things like heart rate, blood pressure, EEG, ECG. Those can be with telemetry implants where you are putting that part in, and obviously you wouldn't need a tether for that, but you do for the dosing and sampling that you want to do concurrently with it. Or it can be for a direct electrical lead, which is one of the things that Dr. Cirrito will talk about in just a moment. So why does all of this matter? Putting this system in place lets you capture data that isn't possible with other methods. So I'm going to refer back to our team that did the work to validate the Raturn system for their automated blood collection and telemetry. The reason that they thought this was so essential is because prior to that, they had two satellite groups, one for safety pharmacology and one for the blood collection. And what would happen is that sometimes you would have an outlier, you would have something happen that you couldn't quite explain, and you didn't have the data about what drug was in the system and when in order to correlate that data. So putting these things together in that scenario meant that they were able to capture data about what was happening in the drug dosing, what was simultaneously happening in the body response to that. Additionally, you're using fewer animals a lot of the time. So this won't be true 100% of the time, but the ability to combine those types of methods together means that you can eliminate satellite groups and put all of it into one animal at one time. The animal becomes its own control and is able to produce more data so that we're using our animals more efficiently. And then, of course, you're able to collect more data. You saw all the various types of data collection that are available with a system like this. One of the references available in this webinar is a link to our bibliography page. It's a searchable page of publications using the Raturn for the last 10 years. And you can see all the types of data that get collected with a system like this. It's across the board, and we're excited to hear more and more ideas about how it can be put to work. That's it for me. I hope you have a better understanding of the system, how it works, and why it can make things better for you. And now you get to hear the super fun part from Dr. Cirrito about how this actually works in real life and what impact it's had on his studies.
Thank you so much for your presentation today, Candace. John, feel free to take it away whenever you're ready.
Okay, thank you. Candace, thanks. That was a good introduction to the Raturn system. So I'll talk about a lot of the work we've been doing looking at measuring proteins over time as well as neural activity in mice in the setting of Alzheimer's disease. So for those who aren't familiar, Alzheimer's disease is characterized by a couple of pathologies, one of which is amyloid plaques or A-beta that builds up in those plaques. Here's a picture of a stain of a human brain that's been stained for plaques. You can see plaques littered throughout the brain. The amyloid beta peptide that makes up those plaques are permeated in neurons, then secreted into the interstitial fluid as a normal process that happens in all of us. where it's normally a single monomer or small oligomers. At some point in the pathogenesis of Alzheimer's, A beta within the interstitial fluid aggregates and forms these toxic plaques or toxic oligomers, which is concentration dependent. So the higher the concentration in the ISF, the more likely it's going to form these plaques. So what my group's been interested for the last almost 20 years is to understand this interstitial fluid pool. And it turns out that neurons firing, just releasing neurotransmitter, drives a beta release into the ISF. So we want to be able to understand how the protein's changing over time, as well as how that relates to neuronal activity or synaptic activity in a mouse's brain. So to do that, we've turned to two technologies. One, in vivo microdialysis, which is a technique that's been used for the last 70 or 80 years, usually to measure small molecules in the brain. And I'll tell you a little about that technology and how it works in just a second. But we can essentially measure A beta levels. We've also measured tau or alpha-synuclein every hour over days in a mouse that's awake and walking around. To do that, it requires to have tubing that's connected to the mouse's head to both push fluid into the probe as well as extract fluid from the brain or from the probe, which means, and the Raturn helps us keep those lines connected throughout the course of the experiment. The other thing we want to do is relate how protein levels are changing to neuronal activity. So we've used two different types. We've either done depth EEG, where we put stainless steel wires into usually the hippocampus for us to measure neural activity at the same place that we're measuring A beta levels. We can also measure single evoked potentials, where you stimulate the perforant path, which leads to the dentate gyrus. You can measure individual EPSPs in the dentate gyrus as a consequence of that. For that kind of technique, you need to have wires attached at the same time. In a lot of these experiments, and I'll tell you about some of this, is we do both of these together. So we've got tubing and wires connected. So again, having a system where we can keep everything connected at once has been really useful to us. So the advantage of the Raturn system for us is that we have a direct line going from our syringe pump to the, in case of microdialysis, syringe pump directly to the animal's head, then tubing, and then tubing then goes back out to a fraction collector. Or we have wires are going from the animal's head to an amplifier to be able to measure the synaptic activity. And we've used liquid swivels in the past. When we started doing microdialysis 18-ish years ago, we used a liquid swivel, and they work. We had a lot of problems with clogging and contamination, however. When we're doing peptide microdialysis, we put carrier protein inside the perfusion buffer to prevent the protein or A beta from sticking to the plastics of the tube, from the collection tube, from the tubing, and the probe itself. And we were having the albumin that we were putting in there clogging the liquid swivel after a few experiments. So liquid swivels for us, we just couldn't keep them clean enough and keep them running long enough to be really useful to us. By taking that out of the equation, just having the tubing direct into the animal's head was really helpful. And same thing is for electrical commutators. They're useful and they work very well, but you can't combine that with other types of techniques. Electric commutators are still prone to some type of failure. So getting, for us, the Raturn is certainly a more costly version of getting access to an animal over time, but it's also the simpler route that by taking as many pieces of equipment out of the equation makes things much less prone to failure for us. And we keep these mice alive for 3 to 5 days in these cages during the experiment. And in some cases, we have mice even a week before that habituating to the cage if we're doing behavior studies and things like that, which I'll tell you about. So in vivo microdialysis, the way this works is you implant a probe with a semi-permeable membrane at the tip. Molecules that are small enough to cross that membrane can enter the probe. They run a solution to the probe at a constant flow rate, usually for us a microliter per minute. And you can sample what's inside, what diffused into your probe over time. So in 60 minutes, we get 60 microliters of fluid, and then we then run that on a ELISA or a Western blot when people have done mass spec to be able to see what kind of proteins or what's happening in the brain over time. On the bottom left image, here's a picture of, this is a BASI probe. This probe is a 4 millimeter long probe. We've used probes that usually 2 millimeters, sometimes even down to 1 millimeter, depending on the brain region we're putting it into. But they're reasonably small, and it seems like it would be a tree trunk going into a mouse's brain. It's not quite the case. A lot of times we create some damage going in, of course, but the damage is actually pretty limited in scope in our hands. On the right is a picture of a mouse, and you can see tubing that's going into the mouse, and then tubing that's coming out of the probe that's then going to a fraction collector. And this big black thing attached is a leash, essentially, so that connects to the animal. So as the animal walks, the whole cage is rotating the other direction. So here's an example of what we've done with this, and we measured baseline A beta levels in the hippocampus over 6 hours. Then at time 0, we injected an SSRI, an antidepressant, into these mice. In this case, we gave one of 3 different SSRIs. You can see in the colored lines, A beta levels go down by 24 hours. A beta levels decline by 25% over the course of that time, over the course of 24 hours. The 3 different SSRIs have different kinetics. Some go down fast, some go down slow. I think that's a property of the drug, not a biological property of how serotonin signaling is modulating A beta. But by 24 hours, and all the SSRIs that we've tried, I think 6 different SSRIs now. They're all down by 24 hours. And then after you can continue monitoring the mice after 24 hours, at some point A beta levels come back up to baseline. And again, that varies between the drugs. And again, I think it's a property of the drug that we're measuring. But these mice are awake and walking around the entire experiment. So you can see how much A beta has changed, as well as how fast it has changed, and is reversible or not. The other kinds of things we can do, because we're measuring things over time, we can do combinations of different treatments. So here's measuring baseline A beta levels again, and then pre-treating mice with an inhibitor of either MEK or ERK. These are 2 kinases that we believe are playing a role in A beta metabolism or A beta production. So you can see if you inhibit MEK or ERK in either the blue or the red, the inhibitors alone increase A beta levels. Then at time 6, we injected citalopram, an SSRI. And normally the SSRI would lower A beta levels, but in this case, we've blocked that effect. So we've essentially, as an occlusion experiment, block a kinase and then see if you give another drug and see was that kinase part of the signaling pathway or not. You'll compare that to the green lines where we dose with vehicle and then give the SSRI. The A beta levels go down by, again, 25%, similar to what I showed in the other slide. In the data that I'll very summarize, we've essentially mapped out this whole signaling pathway in a living mouse, a mouse that's awake and walking around over days, to find that it's only certain serotonin receptors that are being active, 5-HT4, 6, and 7 receptors, that are activating protein kinase A, that are leading to MEK and ERK activation, which are then altering A beta generation as a consequence. All this occurring in a mouse that's awake during the entire experiment. Of course, this is all linked to synaptic activity. So you want to know how synaptic activity is regulating A beta levels. So in order to do that, we wanted to measure activity. So what we've done is taken 2 stainless steel wires. And here we've got a metal support pole that's got, and if you look at the very bottom in the red circle, there's 2 little wires. They're actually the recording electrodes that are being inserted into the hippocampus. And then with that connected to a bunch of other equipment, you can measure baseline EEG activity in the upper right. This is normal activity that you'd find in the hippocampus. And then if you infuse picrotoxins, a GABA receptor antagonist that blocks GABA receptors and essentially disinhibits the system, you see spikes in activity as a consequence of infusing picrotoxins. So you can see, again, in a mouse that's awake and walking around, you can see these changes in activity over time in these mice. And of course, we want to be able to measure activity and A beta levels at the same time. So we've taken those same 2 stainless wires, but now attached them to the microdialysis guide cannula. So at the very bottom, you see in the orange or yellow piece, you can see the 2 millimeter microdialysis probe sticking out of the guide cannula. And then we put the 2 stainless steel wires that are going, the tip of them is where we're measuring activity, essentially midway through where we're sampling A beta levels. And on the picture of the mouse, you can see tubing going in as well as the wires connected. So both of these have a direct line to the equipment that we need to be able to run these things without any kind of break in the system. And then you can measure EEG activity in the upper left. If you measure EEG activity and then infuse tetrodotoxin, TTX, it's a sodium channel blocker that blocks action potentials. As you'd expect, activity goes down. On the bottom left, you can see here an example. After infusing TTX for 16 hours, the EEG signal you have is essentially noise at that point. In the upper right, if you're measuring A beta levels during this, over the 4 hours of TTX infusion, A beta levels come back down. and then come back up pretty quickly once you wash away the TTX. There's a nice correlation between the amount of A-beta you have in the ISF and the EEG amplitude that we're measuring. So here I'm having all of this, and you could have done this in separate mice and have one mouse where you're measuring EEG activity, another mouse where you're measuring A-beta levels, and be able to combine that data somewhat. But it's a much more powerful system to be able to combine these two things together to be able to measure them at the same time and make these direct correlations. You can also measure evoked potentials. So here, implanting a stimulating electrode into the perforant pathway. The perforant path leads from the entorhinal cortex into the dentate gyrus. So if you stimulate, you can measure individual EPSPs as a consequence of that. In mice, you generally do this perforant path to dentate gyrus. In rats, because they're bigger, and a little bit easier to work with as far as just the sterics of all of this. And you can put electrodes into CA3 and record in CA1 at the same time. But you can measure individual EPSPs as a consequence. If you stimulate that pathway at high frequency, you can generate a seizure. This is a kindling model that people have generally used in rats. But if you stimulate at high frequencies, you can see this big, on the upper right, this big high amplitude, high frequency bursting of activity. And when you stimulate, A-beta levels go up within the first 30 minutes. And as long as activity stays high, A-beta levels stay high. And when you stop stimulating, activity comes back down, A-beta levels come back down as well. So here's a much more intricate, and here we've got a microdialysis probe in place, we've got a stimulating electrode, we've got a recording electrode, all going into one mouse's head, which I'll admit there's not that much space left to put anything else in the animal's head at that point. But we've got a lot of different wires coming in and out, and tubing coming in at the mouse over the course of 3 days. Then we have what we call the mega electrode, where we're doing the electrode that we can do depth EEG to measure activity around the hippocampus. We've got a microdialysis probe in place that those EEG electrodes are attached to. We've also done combined EMG to measure muscle activity, and 2 cortical electrodes to measure cortical activity. So we can do essentially sleep-wake by comparing EMG and the 2 cortical electrode activity. You can separate whether mice are awake in non-REM or REM sleep at the same time to be able to do sleep studies during this. And here's an example of some of that. We found that when mice are awake, and this happens in people as well, A-beta levels are high. When they go to sleep, when the lights turn on, A-beta levels come back down. This is the black and the open circles. And if you, at the same time you're measuring sleep activity, this is plotting the amount of time asleep. So the amount, when the mice are sleeping less, A-beta levels are high. When they wake up, when they're sleeping, levels go down, and there's a nice circadian rhythm in A-beta levels. What we generally do when we're doing behavior studies like this, or we're doing sleep stress studies in mice, we'll implant the guide cannula 5 or 7 days before we actually want to do our real experiment. Put them in the cage, hook them up to the tether so they habituate to the new cage and the cage rotating. And then 5 or 7 days later, go in and put the microdialysis probe in very briefly while they're anesthetized, and then start our experiment. So we certainly find if we try and put a mouse into this cage without habituating, and we don't see this nice sleep pattern over the course of, I guess, 3 days we're showing here. But habituating for 5 to 7 days brings out this nice circadian rhythm in A-beta levels, as well as a pretty normal sleep cycle. So with that, I'll thank him. I have a large lab that is actually one doing all these experiments, and there's a bunch of collaborators and certainly our funding sources. But I think with that, hopefully we'll have some time for some questions.
Thank you so much, Dr. Cirrito. That was awesome. So we are going to move into the Q&A portion of our session today. All right, let's jump right in. So John, this is a question for you. Can you perform behavior tasks in the Raturn, and which ones?
You can. Certainly, you're limited, and the animal has to stay in a reasonably confined space. And we've done novel object recognition tasks. Novel object location tasks could probably work as well. Beyond that, you certainly can't do water maze and experiment and things like that. You'd have to worry about things that are a confined space, but certainly we have done that in the past. Yeah.
And Candace, this question is for you. Does the Raturn record its own movement?
So the answer is yes. I made the note that the system is not a behavioral collection system, and I'll try and explain what I mean by that because we're using terminology that isn't necessarily commonplace. But we are not able to capture every single thing that the animal does. The way that the system works is that the animal has 270 degrees to move where nothing happens. And it's only once the animal reaches past that point that a flag enters a sensor and that triggers the counter rotation of the cage. So, if the animal's sleeping, which is honestly what they do most of the time during the day, then nothing's happening. You're not able to collect it. Likewise, if the animal is moving in that 270 degree area where it's not triggering the sensor, then you're also not capturing it, even if the animal is moving. So where it comes in most useful is for applications where you want to understand a baseline change in activity. Right, it is very easy to monitor things like a change from activity levels from day to night. You can see that very clearly with an animal, even when it's acclimating to the system. But also things like I mentioned, where we're able to dose a drug, amphetamine or something like that, and of course then watch the resulting increase in activity. So there's a separate module that connects to the system with some software for the activity collection, and it's essentially just giving you data that you're able to break into the time sections that you want so that you can get the resolution that you want for your application.
That's excellent. Thanks, Candace. John, this one is for you. In your experience, do the electronics of the cage create an electrical interference in noise in EEG recordings?
Generally, no. So for EEG, when you're measuring gross activity, we don't have any kind of interference. When we're doing single EPSPs, occasionally we get interference, and we've got another technology that essentially using amperometry to measure oxidation of molecules where we get some interference. And what we've done is made a self-enclosed Faraday cage when we think we've got this issue, so we just want to be careful, and wrap a cage in copper, both the top as well as the bottom and then the sides, to shield that electrical noise. But in general, that's not a problem that we have. But there are pretty easy ways to get around that. If you find that, if your room has a lot of electrical noise or there's something else going on, simply wrapping everything on copper solves the problem.
Okay, fantastic. So this question is from Thomas. What habituation time is recommended when using rats? Candace, maybe I'll start with you, and then John, you can maybe chime in with some experience.
Sure. Yep. So the answer depends a bit on exactly what you're trying to accomplish. So you saw in some of the data that I showed that when you're looking at something very stress sensitive, like the heart rate and blood pressure measurements, then you're going to have to wait quite a bit longer for the animal to get used to the caging. Remember that was the same for any home cage, so it wasn't any different on the Raturn. Outside of that, when you're just kind of doing a study where you're not quite as worried about stress parameters. So for a lot of our customers doing pharmacokinetics with this system, they will typically do somewhere between a 4-hour to 24-hour acclimation period. And so again, it's dependent on the application area that you're using it in. I would say you should I would never recommend going less than 4 hours. I would generally prefer that the 24 hours is kind of the baseline, just to make sure that the animal goes through a day-night cycle and you can make sure everything is working before you dose them the next day. But then, John, would you like to say? Go ahead.
Yeah, it's for us, it depends on what we're doing. If we're just measuring, the effect of a drug on A beta levels, some of my people will do surgeries in the morning and in the afternoon set up the actual microdialysis to let it go overnight. So in that case, there's 4 to 6 hours or so habituation, sometimes maybe even less sometimes. But in those cases, we're not so much worried about the animal's behavior at all. We're worried about some biochemical change going on in the brain that we're trying to measure. For things where we're doing sleep studies or stress studies, we'll have the mice habituate for 5 to 7 days. We certainly know 1 or 2 days they're not ready for that point. We haven't tested day by day to know whether 4 days is good enough or not. We know that 5 to 7 days is plenty. But again, that's where where we think the animal behaving is a critical aspect of our experiment, or we think it could affect the biochemical change that we have going on in the brain. So again, it all depends on the application and what you think you need to be looking at.
Great. This question is coming in from Andy. In general, are you seeing an increased requirement for hands-free data collection and a higher importance on reduced animal handling and intervention? Is this an important factor that might influence data acceptance and success in publication?
It certainly plays a role. And if you're trying to look at stress and how stress could be, and there's a lot of things in our science that stress is impacting, and certainly handling it and being near the mouse, handling it, injecting it, whatever you're doing, is a factor. Whether it's for a lot of what we do, we don't try and minimize the intervention we're dealing with the animal just because it's difficult for us to do that. And I'm not sure experiments are negatively impacted from that. I'm not sure that it's a crazy. It would be the factor that would determine data acceptance or success. Though, if you're studying stress, it wouldn't hurt having less interaction with the animal.
Yeah. I'm going to weigh in this one a little bit as well, because obviously John knows a little bit more about the acceptance and success for publications. From my end of things, what I've seen happening over time, and this is related to a lot of things in our culture and our world, but there's been an increasing growth in animal research to make sure that we are talking about what we do and talking about it in a positive way. And there's a lot more, like just kind of layperson paying attention to what's happening in animal research. And so that's one part of what's driving these changes, but people do seem to be focusing across the board, whether it's at pharma or academia or industry, seem to be focusing more on the impact that humans have on animals in their studies. And so this is something that a lot of behavioral researchers obviously have paid attention to for a very long time. But there is actually a name for the field of study where you look at how animals are impacted in their research by human research design. I'm going to say the name really terribly, but it's like therapistemology. Please forgive me for that, whoever actually knows how to say it. But yeah, so a whole range of people studying now things like how we do cage changes and how that affects the animal stress and how we hadn't paid attention to that and how it might have an impact on data. So yeah, from the publication perspective, maybe not. I don't know if that's come into play too much just yet, but from the general approach that research is heading and the way that we go out into the world to talk about what we do, it's definitely changing.
Okay, great. Thank you both so much for that. A question that we've gotten a few times here is, can you house multiple animals within the system? John, we'll start with you.
No.
Yes. No, it's correct.
Well, in order for the mouse to be, if it's tethered to something, if you have two mice moving in opposite directions and you can't keep them both tethered, so you can have one mouse tethered and another mouse just walking around normally, and that would, I wouldn't see any problem with that. If there's, I guess I can envision a time you'd want to be able to do that for something, but you can only, at least we haven't been creative enough to find a way to tether more than one mouse and not make the whole thing go haywire. So in general, I would consider it really one mouse per cage. Yeah.
Okay, great. Then maybe Candace, maybe you could tackle this one. How would one utilize this system or this technology at scale? Is it possible?
Yes, it is. So this, I think, kind of feeds into that previous question about being able to do more than one animal. Of course, you guys would all be looking at this and wondering how you might fit it into your lab space. And like in any lab space is an issue. But that's part of the reason that we have the different configurations of the system, so that whether you're kind of putting it on shelving units or putting it on a benchtop, or whether you want us to create some kind of custom shelving system, we're able to help with that. Really, the truth is that in terms of technicians or students that are interfacing with the system, once you're able to set up once you have your experiment down pat, it's typically relatively quick to set up multiple stations. Most of our customers have somewhere in the neighborhood of 8 or more systems in their lab, so that they've got enough to run, reasonable numbers of animals in their studies at each segment. And maybe, John, you want to chime in and if you want to, and let us know kind of what your lab is set up like and how many you've got.
So we generally, once you're setting up one, it really is, it's not much more effort to set up more than one. And we generally will set up either 3 or 4 mice at a time and then do microdialysis. From my own experience, and I think from most of my people, that doing more than 4 at one time takes a lot more brain power than it's worth, and is more prone to failure. Doing up to 4 for me and I think for us is takes about the same amount of effort and success rate. We try not to scale up much more than 4 at any given time if we can avoid it. But that said, and we've got, I think, 16 Raturns now. So we can set up multiple, on Monday we'll set up 4, on Tuesday we'll set up another 4, and do back-to-back experiments that way. But 4 is our limit in brain power.
That's awesome. Great. Okay. Well, I think in the interest of time, we'll take this question as our last question, and we'll save the rest for our Q&A report, which will be available in the next couple of weeks. This one's from Greg. Is there a reasonable way to monitor timing of food intake when the mouse is in the system? John, in your experience?
We generally put food in the bottom of the cage. We don't have a good way of doing that. We don't, except and we could take the food out, and weigh it after the fact. But that.
Candace, do you have anything to add?
I will say. Yeah. Reasonable way, yes. Excellent way, no. I mentioned that we have the metabolic cage so that you're doing urine and feces collection. That type of cage uses a slightly different design than the one that Dr. Cirrito uses in his lab. The problem here is if you're doing anything with a head mount, you don't want them to have anywhere where they can get that caught. Right. So the cage that is best for measuring food intake is not best for using a head mount animal study. But if that's not what you care about, if the metabolic collection and food intake is what you're paying attention to, and you're doing infusion or something like that, then it is relatively easy. The food hopper is on the outside of the cage, and the animal has access to it, but it's relatively easy to kind of take it off, put a slat in place so that it's closed while you weigh the food, or you can just take it out by hand. So it's not like automated measurement or anything like that, but it is accessible and relatively easy to do when you've got that kind of cage set up.
Perfect. Well, thank you both so much for all the fantastic information, both in your presentations and as part of this Q&A session. And thank you to the audience for taking time out of your day to attend this webinar. We hope you found the information presented both educational and applicable to your research. Of course, a very big thank you to our sponsor, BASI. Again, the slides and recording of today's webinar will be available soon at InsideScientific.com, and a Q&A report will be posted in the coming weeks, including answers to all of the questions that we were unable to address today. Look out for an email or two regarding those resources in the near future. In closing, thank you again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a great day, everyone.